On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
This episode of Life Science Success is brought to you by 5280 Life Sciences Consulting. If you need help setting your strategy, meeting your operational goals, or you are selecting and implementing critical technology in your business, we should talk. Visit 5280lifescience.com to schedule a meeting and find out more.
So today I'd like to welcome to the podcast Michael Fero. Michael is the founder and president at TeselaGen, a biotechnology company in California. Welcome, Michael.
Hey, Don. Thanks for having me.
Yeah, so would you mind just please tell the listeners a little bit about yourself and your career? We'll cover a little bit of your past as well as what you're currently doing.
I have a bit of an oddball career. My academic background is as a particle physicist. So I spent many years doing experimental particle physics at MIT and CERN and Stanford, where we measured what's called the Weinberg angle. But in the late 90s, I got intrigued by biology and realized how little I knew about probably the ultimate complex system, and started to migrate away from physics and into biology.
So as the founder and president of TeselaGen, what does TeselaGen do? And maybe just tell us a little bit about their history as well.
We started Cecellogen about 10 years ago. As a part of my migration, I wound up working with a guy named David Botstein and also a guy named Pat Brown at Stanford. And they also introduced me to Lucy Shapiro. And I started working with Lucy and Harley McAdams on systems biology questions in microbial systems. And that led to working with some cool guys, Eduardo Ablik and Nathan Hilson, who joined me when we started Cecellogen about 10 years ago. And our goal was to really transform the way informatics is done in biotech companies, in particular companies that start from a fundamental DNA or amino acid standpoint, and then either modify microbes or build molecules de novo.
Okay. I've seen quite a few articles about TeselaGen, and you talk about the machine learning aspect as well as the synthetic biology. What is this, and how would you say that a pharma company might benefit, from using your technology and say their next drug discovery?
That's a very good question. I think it really comes down to the fact that, from my perspective as a physicist, biology is not immediately amenable to a physics approach where you have an underlying theory, you can develop a mathematical model and simulation and then go from there. So that works well for electrical engineering, chemical engineering, mechanical engineering, aerospace. But when you get to biology and the fact that it's a complex system, you don't always have all the necessary information to build those kind of fundamental models and do simulation. You can, don't get me wrong, you can do some very good modeling exercises and docking and so forth, but what that really gives you is something that looks like a space of possibilities, a hypothesis space. And I think you should, in fact, this is actually how it's done in biology, is you select or sample or screen from a vast hypothesis space and find the ones that work. And that's the genesis of everything from genetic selection to high throughput screening. And so we try to make that very intelligent. Biology is this big black box, but it has definite inputs and definite outputs. It has genotypes and phenotypes. So there's definitely a relationship there. You just have to model it and understand it. So our approach is to do that kind of model building purposely and early to help companies start to build a model about where in a hypothesis space they should actually look. And depending on how well you've been able to figure out what your hypothesis space is, you may have 10,000 or 100,000 different places to look, and you want to do that very efficiently.
Okay. The question I would have is, where do you typically see that TeselaGen gets most involved? Is it early, or is it late drug discovery, or is it somewhere in the middle?
I would say it is somewhere in the middle. Typically, the scientists who have done the basic R&D on a particular indication, and they have their target and they have their candidates, they have already generated a hypothesis space. Often, though, it's quite large, and you have to be able to navigate it, map the contours of that space, which is multidimensional, and try to find the optimum within it in a very efficient way. And because it's a multidimensional space, just doing a single assay and then sorting it in your spreadsheet isn't going to work. You have several parameters that you have to optimize at once, and that's where these nonlinear methods like machine learning can really be helpful.
Okay, good. And then you talked about this just for a brief second. So Cecellogen offers a laboratory operating system and then also high throughput workflows. Can you explain how this is done?
Yeah, it almost came as I wouldn't say an afterthought, but a necessary infrastructure that you have to build in order to enable this kind of data-driven discovery, if you will. We know that there's a relationship between genotype and phenotype. We know that we can design experiments and we know that we can collect data, but there's a middle piece, which is actual construction of the molecules, which for us is important to control as well, because it's a very common source of systematic error. So the two kinds of error you have to control in a machine learning exercise are the sort of statistical and systematic errors. You can control the statistical errors by doing the right design of experiments and having a properly designed experiment with enough data. You can control the systematics by tracking and orchestrating the entire molecule production process in a very rigorous way, which means full tracking and not relying on humans too much for things like data acquisition. And then finally, you want to acquire that data like I said, as automatically as possible, and then condition it, normalize it, and feed it into your model for model building. So yeah, we had to build the infrastructure in order to enable this paradigm of data-driven discovery. So it turns out we sell a lot of just the infrastructure before people are ready to do machine learning, but machine learning is the goal.
So people would come to you and then they would say, hey, look, I want a limb system, more or less. And my goal is at the end of the day to use machine learning. But for now, I'm just going to collect information in that. Is that how you typically see your operating system getting sold into facilities?
Yeah, it's a 4-part system. We have a design module, a build module, which is the limb system. What we call the test module, which is really a data lake, a place to store high value data. And then we have the discovery module, which is our implementation of our machine learning models. And they'll pop in at any one of those 4 places. If they need help at the bench designing experiments, they may go with the design module. If they have data and they really want us to look at it super bad, they'll say, okay, let's go right to the discovery module and see what can help you generate some ideas. So it doesn't matter too much how they come into us, but we give them sort of multiple entry points. But ultimately, we do have an integrated system. So if they want to do the whole thing soup to nuts, they can get a pretty impressive system for a fraction of what it would cost them to do it themselves.
And so the types of things that you work on overall, are these people that are still primarily screening with HPLCs, or are they using next generation sequencing to model things out? I'm just curious in terms of the types of equipment that they're using to put information into the LIMS portion of the modules, if you will.
Yeah, I think it's all of the above. Design the module is something designing the screen is something you do at your desk. So that you're in the design module, you're sitting at your desk and figuring out what you're going to do. We can generate protocols automatically and send them to the build module, which is the LIMS system. So now you're off your chair and you're over at the bench and you may be firing off a work cell or robot or doing it by hand to build your molecule or make your modification to it. Along the way, you're going to be taking data for quality assurance purposes, and that may include sequencing things and any number of other assays. But there are going to be a few particular assays towards the end of this process, which are the critical ones which capture the important phenotype you're trying to design for. So yeah, a certain set of data goes into the system as just QA data and helps you improve your process. But we're not stopping there. We don't just want to help improve a process, make it more efficient. We're trying to actually discover something. So it's the final assay data is very important to get into the system and have it secure and safe, and then to transform it, normalize it, filter it, so it can be fed into the machine learning model.
Okay. And then once you put it into the machine learning model, are the answers relatively quickly produced, or does it take days and things like that? I guess the one experience that I've had with this before is that within next generation sequencing or NGS, you put in your information and sometimes it could take hours or even days potentially to output some of the information. So just curious, how long do people normally wait for answers?
Yeah, that depends on how well developed the model is. It's virtually instantaneous if your model is already developed. But if you're just taking initial data and starting to build a model, that could take weeks or months to get that first model built and validated. But once it's built and validated and deployed, then it just gets better and better. It doesn't take much time at all. I think you and I talked about high throughput sequencing workflows and stuff. Those are complex workflows, which still people don't have the best solution for. But the more automated they become, the easier it will be for people to get useful results from them. And there's no physics reason it can't be much, much faster than it is. It's mostly just information flow. We don't have good connectivity often in these information workflows. So that's something everyone struggles with, including us. But we think we're doing a pretty good job on our part of that puzzle anyway.
It seems like whenever you really want to make a decision about a biological or biomolecule, your system would really help to quickly sort through decisions for the best path. Your operating system and the other modules that are associated with it, could it help with things like COVID variants and how to best maybe treat for other things that we're seeing come through the challenges with COVID?
I think so. We wrote what I thought was a great proposal to the National Science Foundation, but apparently there were like 1,000 other proposals written at exactly the same time last spring. So we did not get a lot of love from them. They just pushed us off to the future. But I think in the future, what you saw with COVID is the fact that if you put enough energy into it, you can get something developed really quite quickly. And that just speaks to my earlier point about if the effort and the information science is there, you can get things done quickly. And I think that's true with vaccine development. In some sense, vaccine development has a lot of elements that are a bit more mechanistic. If you do step 1, step 2, step 3, step 4, forgetting clinical trials for the moment, you can go from raw sequence to vaccine candidate fairly automated way. So there's no reason we can't create systems that are much more automated, in particular for vaccine development, which is a bit mechanistic compared to, say, trying to develop something against cancer. I think you can. I think you can both build vaccines much more quickly and in a much more automated way, almost like just a machine that you put the sequence in at the front and you get the vaccine out the end, and you could build adapted variants of that vaccine fairly quickly as well. So yeah, I would say so.
In terms of the use of the operating system and the machine learning ends of it, how hard is it to use? And are there consumers of this that actually use it themselves, or do they always come back to you to help build everything out and your company does everything more or less for your clients?
You just download it onto your iPhone and that's it. You're on your own. It's not quite an iPhone app, I admit that. We like to think we have a nice interface. It's getting better and better all the time. There's a bit of setup. The dirty word in enterprise software is customization. So we call it configuration. And so we have to distill a lot of requirements from a very heterogeneous environment. R&D departments are everyone's doing something different. So we have to distill a lot of requirements into something that can be used by anybody and then configured either through the admin panel or through us tweaking something. But yeah, we strive for as little of that as possible.
Yeah. And how long does that process normally take? So if I came to you today and I said, hey, I have something that I definitely want to use your operating system for, what does it normally look like for most of the clients that you work with?
That's a very good question. So we have a pretty well-developed process. It's taken us a few years to get there, but we do usually what's a discovery process where we do what we call a proof of concept, where in a matter of a few weeks, we'll quickly capture the pain points and the workflow requirements for our customer. We'll see what we can configure in the system so they can get started right away, what they can do right away, and what gaps might exist for which we might have to develop some new tooling. And that takes 4 to 6 weeks, and then they're off and running.
I have not lived through the entire drug discovery process on any drug. Typically, I've dealt with clients who are on the back end of things. I've also interacted on the front end of things whenever I worked for GE Healthcare, and we did a lot of the implementation of the HPLC equipment that we had. And so I've seen both ends, but never had to live through the entire drug discovery process, of which I hear it can be a decade potentially, and 4 to 6 weeks is probably not as significant as it could be in a 10-year life cycle for a potential drug discovery. So that's good. I like to ask 3 questions of every guest. And so what inspires you?
Big wave surfers. Peter Mel just caught wave of the winter at Mavericks a couple of days ago. It was really inspiring. I grew up in California, and my mom was a dancer. My dad was a test pilot. His weird upbringing. She turned us into figure skaters, and we live like blocks from the ocean. So as soon as I could, I rebelled and got into the ocean and started surfing. And I've been pretty stoked about that ever since. So, yeah.
And you still surf? So you get out there, and will you get out there in the winter at all?
Oh yeah. Yeah.
Okay. All right. Would hope that it's in a full body suit.
Well,
it's probably quite chilly even in your ocean. So
it's called a 4-3, 4 millimeters on the chest and 3 millimeters on the arms and legs. Yeah, it's warm enough.
All right.
It's not too bad here.
And what concerns you?
What concerns me is, oh boy, what to be concerned about? My God. I don't know. Scientific ignorance concerns me. The ability to naively believe what you might see on a social media post or a Twitter feed and not really critically challenge it. That's pretty concerning.
And what excites you?
I guess should have saved that for the surfing one, but
you can repeat it because your signal kind of faded on me.
Yeah, the surfing does excite me. I have a lot of fun with that, but I guess I have to be a salesman here and say the system that we're building is pretty exciting. And I think after a decade, we're finally getting what the investors like to call traction. And people are actually seeing that this is the right approach, to take a serious informatics approach to this pipeline and not try to just paste a bunch of stuff together in an ad hoc fashion. But yeah, it's pretty exciting.
Yeah, it's interesting because I get these phone calls now where people want to know, so what are the key things that I'm hearing in life sciences? What are the most exciting things? And to me, it's between next generation sequencing, what we're doing with CRISPR, what we can do with artificial intelligence and machine learning, I think are all at that right point right now. And there's a lot of just great things that are happening with the different companies that are out there. Couldn't agree with you more on that one. So Mike Fero, thank you very much for being on the podcast. Thank you for spending time with me today. I greatly appreciate you explaining a little bit about Cecellogen and what you do there. And I wish you all the luck.
Thank you, Don. I appreciate what you're trying to do here. This is exactly what folks need, is good informative content so they can learn about what's happened in the world.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.